Hello, and welcome to this scientific digest with the topic From the Clinic to Translational Research. My name is Caroline Ospelt, and I'm a group leader at the Center of Experimental Rheumatology at the University Hospital in Zurich, Switzerland. I'm actually quite happy that EULAR asked me to give this scientific digest because I gave an educational lesson about this topic 2 days ago for the young rheumatologists. And actually also yesterday, there was a very interesting session called From Bedside to Bench, Unexpected Results of Clinical Studies that Changed Our Understanding of the Disease. So I really think this is an important topic to discuss and to think about because it takes a bit around how we normally do research. So often what we do is we come from the bench, we have an explorative study where we look for targets, where we identify targets or biomarkers, and then we go to the clinic and test whether these are valid. Sometimes we forget to go back. So what we really should do is we look to the clinical data because this is our reality test. There we actually see whether our results were or our analysis was correct or not. And then we see where we have to go again and where we have to have a closer look and where also the gaps in knowledge are. So where can we really make a difference with research, where lie the problems, and where can we still improve the therapeutic options and the life of the patients. So in the next couple of minutes or so, I want to address some of the problems that I think come from the clinical data and that we can address in translational research. The first topic that I want to address is why do not all of the targets that we found in translational research and we verified there work at the end in the clinic? So we have some examples that worked really well. And I think in general, in rheumatology research, we did very, very well in identifying targets and in treating patients. For instance, TNF is a good example. Anti-TNF blockade works very well. And I think we were lucky that this was one of the first targets to be tested because it really gave the community the trust and the confidence that this approach is correct and that we can targets that were identified in basic and translational research to the patient that this would make an effect. Another example is IL-6 blockade. At least in rheumatoid arthritis, IL-6 blockade works very well. We can block the cytokine, we can block the receptor, we can block the signaling pathways with the JAK inhibition, and we all see a very good effect in patients with rheumatoid arthritis. However, there were some examples where actually it didn't turn out as we expected from the research side. One example is IL-17 blockade. So in preclinical model, it looked as if IL-17 would be the ideal target in rheumatoid arthritis patients. Unfortunately, then, the first clinical trials really showed that the effect was modest and not what we expected at all. The same is true for IL-23, actually. Also there, in rheumatoid arthritis patients, we didn't really see an effect, even though a preclinical model would have suggested a strong connection of IL-23 with IL-17 and a strong influence of these two in rheumatoid arthritis. However, we were not completely wrong, because even though in rheumatoid arthritis, these two cytokines seem not to have the role that we expected. At least anti-IL-17 worked very well in patients with ankylosing spondylitis. And also there again, we see that IL-23 didn't work, even though we thought there is a strong connection between IL-23 and IL-17, but then IL-23 works very well in patients with psoriasis arthritis. So what we can learn from that is that maybe in basic research or in translational research, we don't have the models that really show us which cytokine would be active in which of the different chronic inflammatory arthritis that we see in the clinic. So actually, what we have to acknowledge is that the preclinical animal models, for instance, that we have, they are models for inflammatory joint disease. But we should be careful and we should not call them RA models or SPA models, because that's not actually what they are. We can mimic a chronic inflammation in the joint or acute inflammation in the joint, but obviously we cannot mimic the different diseases that we see in humans. So what to do to come to this point? So I think what is really important is that we increase our effort to learn more about the pathogenic pathway in each of these different chronic inflammatory arthritis. Because nowadays, actually, I have to say the bias lies strongly in the research of RA. So we have a lot of knowledge about what is going on in the peripheral blood, in the immune activation, in the synovial tissue in patients with rheumatoid arthritis. However, we have much, much less information what is going on in the joints of the patients that don't have RA, but have another chronic inflammatory disease. And we see clearly from data from the clinic that there are differences. And we have actually to see that also concomitant symptoms like gut inflammation, skin inflammation, enthesitis that we see in these patients, they have different pathways affected. And we need actually to go back and check for all of these different arthritis and all of these different disease manifestations to really tease out which pathways are activated there. So I would strongly suggest that we go a bit away from RA, even so I think RA is a very important topic, but also in the clinical trials, what usually is done, we test the drug in RA patients, and then we go to other diseases, but that we get a bit more aware that each of these chronic inflammatory arthritis is a different entity, and that we have to acknowledge that different pathways and different hierarchies might be in place there. So this was the first part of the scientific digest. I have a second problem that I want to address, but for this, we'll take a short break and come back afterwards.
Since March 2020, we've had to adapt to home working, remote health appointments. Too often, we could only get support digitally. It was not possible to travel or meet friends the way we enjoyed before. We've missed the lack of personal interaction. The corona pandemic has turned all our lives upside down. We have to be aware that while many aspects of digitalization have been welcomed, for some people, it is a challenge to navigate the technology. Especially for people with a chronic condition, it is essential that they are able to use the digital options that may improve their quality of life. At EULAR Power, we strive to raise awareness about the impact of living with RMDs. This is why we invite people to share their experiences. So thank you to all those who contributed to the 2021 Edgar Steiner Prize competition. I hope you all enjoy reading the essays as much as I did.
In what has been a very difficult year for everyone, EULAR Power is delighted that people with rheumatic and musculoskeletal diseases, RMDs, took the time to take part in our annual Edgar Steiner Prize competition. Each year, we set a team asking people to explore issues relating to living with RMDs. As the impact of the COVID-19 pandemic has persisted, the manner in which healthcare and support has been delivered has changed significantly. When we set the topic, we did not realize how truly resonant the theme "How digital solutions benefit my life as a person with an RMD" would be. I'd like to thank the judges who gave their time voluntarily to read through every single competition entry. It is a task everyone always enjoys, but it comes with big responsibility. The jury was united in choosing Stine Björk Rondon Jeffson's essay as our 2021 winner. A description about feeling connected resonated with everyone. Enjoy reading the 2021 entries. I know I did.
Hello and welcome back to the second part of the Scientific Digest with the topic from the clinic to translational research. So in the first part, I was talking about that we have to learn more about the differences in the pathways between the different inflammatory joint diseases to improve the efficiency of bringing new targets to the patients. Now, in this part, I want to address another topic, and this is why don't we get the full treatment response? Because when we look at all the clinical trials actually in chronic inflammatory arthritis, what we see is that usually we get an ACR20 response. So that means 20% improvement in signs and symptoms of the arthritis in around 60, 70, or maybe 80% of patients. So this is actually in a one-way great because we reach a big part of our patients with the drugs that we are using. But then we have to consider that 20% improvement is not really tremendous. I mean, I don't think people are happy with only 20% improvement in signs and symptoms. On the other hand, we also measure ACR70. So that is 70% improvement in the signs and symptoms of disease. And I think this really makes a substantial difference. And some people might even have more than the 70% improvement. So that is a great effect for each individual patient that has this response. But unfortunately, what we reach is like 20 or 30% of patients that are treated that get this response. So I have to say we are used to this data because, as I mentioned before, these are the usually data that we get with actually all our therapies that we are using more or less in all the different arthritis that we are testing. But to be honest, as a patient, this would not what I would expect. So what I would expect is that it's guaranteed that I get a substantial response to the drug that I'm taking, especially because some of these drugs actually also have severe side effects. So why is it that we always seem to have a ceiling or plateau and we cannot get over this? So why don't we have any treatment that actually can reach an ACR 100 in a substantial amount of patients? Now, this is tricky to answer, of course, but it might be that it's a bit the approach we are taking, because as I said, in preclinical analysis, what we are looking at is a joint inflammation. So the most targets that we actually today are using are targeting to suppress this immune response, to suppress the inflammation and the immune cells that are activated. So either it might be that we need a combination of these different targets, because when we block one, it might be that the other target actually can take over. But it might also be that we have to move a bit away from this classical thinking that only this immune activation is having a role in this chronic inflammatory arthritis. So maybe we have to look a bit for other compartments that we can target. And actually, for some years now, there were some studies going on in several fields that showed that the microbiota, so the intestinal biota, can have a great influence on chronic inflammation. And also at this EULAR, there was a nice abstract presented to show the first analysis of microbiota transplantation, so a therapeutic approach to modify the microbiota in patients with psoriasis, psoriatic arthritis. Unfortunately, this trial was negative, but I think this gives us a good idea that we might think a bit out of the box and use different treatment approaches. Another approach I think that we can take and that gave us the opportunity in the last years is that we have to look into the tissue and see what is activated in the tissue. Usually, we measure immune activation and cell activation in peripheral blood, because that's much easier accessible. But in recent years, it was a great progress that we now use often ultrasound-guided synovial biopsies. And this gives us the opportunity to directly look into the tissue and see which cells are actually activated in the tissue. And it might be that these are different targets and that these are very good targets. So also in the plenary abstract session, there was a nice abstract showing that when you compare the immune cell activation in the periphery, it's quite different to what you see of the immune activation in the synovial tissue, so in the joint. That means that this could open up new targets of activated cells in the joints, and not only in the joints. There was also a study showing that in salivary glands in Sjögren syndrome, there is a specific activated tissue-resident T cell, so a tissue-resident immune cell, that if we can target this might have a beneficial response. So in the second part, I really think we should go a bit away from the classical immune suppression that we use up to now. We should be open for new approaches. We should look into the tissue and maybe target tissue resident cells, also stroma cells like synovial fibroblasts or endothelial cells. And with this thought, I leave you for the third part of the digest, and I come back after a quick break.
EULAR guidance for patients with rheumatic and musculoskeletal diseases about COVID-19 outbreak. Do not stop or reduce your immune suppressive medication at this time, unless your physician tells you otherwise for a specific reason. Check with your doctor or rheumatologist if your routine rheumatology appointment is essential, or if it can be safely delayed, performed by telephone or some other remote device. If you have symptoms, you may need to be tested for coronavirus. Follow the national advice for access to health care. If you are found positive for COVID-19, please discuss with your doctor or rheumatologist as to what to do with your treatment. Undertake or complete vaccination protection according to EULAR recommendations and/or national guidelines. Look for the advice offered by your national public health care system. Coronavirus. Protect yourself and others.
Welcome back to the third and the last part of the Scientific Digest, from the clinic to translational research. So in the first part, I was discussing that I think we have to learn more about the differences in the pathways between the different inflammatory joint diseases. In the second part, I was pointing out that maybe we have to add a therapeutic approach that has a different target than the classical immune cells and immune response to improve the response rate and the efficiency of the therapy. Now, in the third part, I go a bit in the same direction. And this is, how do we find the best treatment for each individual patient? And this is, of course, the topic of personalized medicine. And I think in the last year, this is something that was really the aim of everybody working with patients. What we want is that every patient gets an individualized treatment fitting to the pathways, to the symptoms, and to the pathways that are actually activated in the different diseases. So to do that, we really have to find out what makes our patients different. How variable are they? So for people working in clinic and working with patients, for them it's quite clear that somebody with the diagnosis of RA can look quite different to somebody with the same diagnosis. So RA can have a very destructive, very fast course. It can be that some patients, they don't have such a fast course of disease, but they might have comorbidities. Some might respond better to treatment. Some might respond less to treatment. So all these patients are quite heterogeneous. But the task of the research is now to find out what really makes them different and how can we stratify these patients into similar subgroups, that all the patients that look similarly on the pathogenic pathways that are activated can be grouped together and maybe treated together. So research has many approaches to do that. And as I said, the story is not necessarily new. So for a long time, we already tried to stratify the patients into more logical groups so that we can treat them better. So one approach is genetic, of course. So the plan was that we can measure the genetic background and then stratify the patients according to this genetic background. And this would give us information about comorbidities that the patients would develop or about the response to therapy. Now, in general, I have to say, the data that came out from this was not that we can use this in clinic already. However, some of this data is still very interesting, and we should really follow this path. So, for instance, in the plenary abstract session, there was a nice abstract presented that showed that patients with a MUC5B genetic variants were at a very high risk to develop interstitial lung disease within rheumatoid arthritis. So that means when you were a carrier of these genes, apparently the risk to develop lung disease is much higher, not only when you have RA, but actually also when you don't have RA. So this gene somehow seems to be connected to interstitial lung disease. Now, in future, this really could be used that we screen people for this gene, and when they have this risk variant, we can either already look whether the lung is still healthy, or whether we have to put extra care of this patient, maybe some preventive measures. Or what we can also do is that we look what this gene actually does. So which pathways are activated by these genes? Maybe we can find some therapeutic target that we could use to actually treat these patients that specifically are at risk to develop interstitial lung disease. So in theory, this genetic risk factor could really help us to find better therapies and to stratify the patients better. These genetic risk factors have also tried to be used to stratify patients according to treatment response. So we thought it could be helpful to find genetic background that would predict whether a person would respond, for instance, to anti-TNF treatment or not. And it turned out that it's not so easy, but we can combine different parameters, and that makes the prediction score much better. There was also abstract presented and data presented in this meeting, but also before, people tried to combine genetic data with clinical data, and that could see that this really improved our ability to predict who would respond and who would not respond. So unfortunately, also this approach is still far from being taken into clinic, but it shows us that maybe the way to go is that to combine different parameters that we already know. So we have a lot of genetic data, we have a lot of data that analyze the predictive and prognostic utility of clinical data and demographic data. We have protein biomarkers that we could measure in the blood. We have gene expression analysis that shows us which activated pathways might be connected to response or non-response. And what we really have to do now is to combine all of this data. It's maybe a bit like Mark Pollock so impressively mentioned in the opening session. We have to work together and we have to be collaborative and put all this data together. And the new computational tools that we have, like machine learning approaches, really allow us to do that. So what we have to do is to combine all this data and then see if this would help to stratify the patient and to find the activated pathways in each of the different patient groups. And this, again, could also help you to find the fitting treatment. Now, another revolution actually in research technology was the single cell analysis. And also this helps us greatly in stratifying patients. Now, this technique really increased the resolution that we have now to look at the different players that are active in the disease. So you have to imagine that it's like you use a camera and the focus is not set correctly. So if there's a blurry picture, it's really hard to differentiate if different people on this picture will have, for instance, blue eyes or brown eyes, or in how far they look different. So this is with what we were working in the last couple of years. Now, the single cell resolution actually helped us to focus this picture. So now we can zoom in and we can exactly say, this person has blue eyes, this person has brown eyes, and so forth. So with a single cell technology, we can increase the resolution so much that we can maybe see small cell populations that are only active and only present in some of the patients. And this might also give us new targets that we can either develop for these patients, or maybe just repurpose because we see that maybe pathways are activated that we already can treat. We just maybe did not always use them for the right patients. So all in all, I think we have a great deal still to do, but we live in a very, very exciting time. We see all this clinical data, and I think we improve the life of our patients a lot, and we have some dark spots where we still have to go back and look how we can increase the knowledge in this and go back to clinics and see if this was worthwhile. And I hope that in the years to come, we see a lot of data coming with these new technologies and shedding a bit light into the dark areas that we still have. So with this, I hope you enjoyed a bit this scientific digest. You could learn a bit something. This scientific digest also will be available on the platform and on the YouTube channel in the coming days. And if you want, you can contact me on my Twitter handle. You can discuss with me and give me some input about this topic. I would really be happy to hear from you. So have a nice afternoon and enjoy the rest of EULAR. Thank you.